Good morning to all of you. We have amongst us, we have the opportunity to listen to Professor Ahmed Moustafa. He's a senior lecturer in the Cognitive and Behavioral Neuroscience at Marx Institute of Brain and Behavioral, and also at the School of Social Sciences and Psychology at University of Western Sydney in Australia. I think I would request him to give a self-introduction and tell us about your work on Parkinson's disease. Thank you.
Right. Thanks, my Vice Chair, for having me here. So briefly, I'm originally from Egypt. I did my undergrad in mathematics, computer science, Cairo University. I moved to the States. I did PhD, postdoc at the University of Louisiana, University of Arizona, and then Rutgers University. And then I got a faculty position in Australia, and I have been living there for 3 years. So I've been at the University of Western Sydney, almost for 3 years now. So most of the work I do is actually on Parkinson's disease patients. So I do work called computational neuroscience modeling work, in addition to also experimental tests, mostly behavior testing on patients, as well as also sometimes fMRI studies. So if you guys have any questions, feel free to interrupt at any point. Okay. So we'll try to take about maybe for about 40 to 45 minutes, tell you a little bit about my research, and then I think we'll have some time for questions. All right. Talk about a class of computational models known as reinforcement learning models, and we have been using this to simulate data we have from Parkinson's disease patients. So one project we're working on is called freezing of gait. So the gait problem we have in Parkinson's patients. Another project something called impulsivity or impulse control disorder. It also happened in Parkinson's disease patients because of the medication that they take. Another one also do have short-term memory, or what's known as working memory in Parkinson's disease patients. And at the end, so if we have some time, we'll talk about deep brain stimulation. So are you guys familiar with deep brain? Who's here familiar with deep brain stimulation? Are you guys familiar with it? Anyway, so it's a surgical method for treatment of many psychiatric and neurological patients. So often when medication doesn't work very well, or the medication can cause some problems after some years, people will switch to deep brain stimulation. We do a surgery, you open the brain, you stick the electrode in different part of the brain, depending on the disease people have. It has been used for 20 years for Parkinson's disease patients, and over the years also for depression, anxiety disorder, schizophrenia, and others. And it's under clinical trial for other diseases. And I know there's some places in Kerala that are doing this. I think in the capital. I think I don't know how to pronounce it, so I'm not gonna say it over. It's like a very long word. Anyway, so it has been happening also some other part around India as well, and many other part of the world. So I'll talk about this at the very end. All right, so what is Parkinson's disease? Parkinson's disease is a motor disorder, usually happen in old age, but it can also happen in younger age. But it's not very common in young age. But very famous example of people get it when they were young, Muhammad Ali and Michael J. Fox. But again, this is not very common. And the main symptoms people use for diagnosis are this cluster of symptoms. So like rigidity, akinesia, which is basically the term that really like Parkinson's disease patients have very stiff hands. If they try to move their hands back and forth, it will almost like it doesn't really move. So it's called sometimes cogwheel movement. It's not very continuous. It's making move and stop and keep drag it and kind of stuck again and so forth. And it's again cogwheel movement. Akinesia is just a term for difficulty initiating movement. It takes them time to initiate movement. If a patient is sitting down as going to walk, it will take few seconds to process the information to make it the switch a different motor plan. Resting tremor is just basically the tremor patient have at rest. If they're sitting down or don't do anything, they start shaking. Often when they start moving, the tremor goes away. But so that's different from action tremor, which also can happen sometime in Parkinson's patient, but not very common. But so the different kind of tremor we see more often in Parkinson's disease patient. And bradykinesia is a term for slow movement. If Parkinson's disease patient doing some tasks usually take them longer time than a healthy participant, than a healthy person. Okay, so this is actually the main symptom we see in Parkinson's disease patients. Actually, these are not everything. They also have balance problem, posture control problems, handwriting problem. You have sometimes flat face. It's a very common symptom we see in Parkinson's disease patient as well. Actually, the most difficult problem also, they have also swallowing problem. It's a motor process, and actually it's one of the most dangerous one because they can't swallow very well. It can lead to death sometimes. And we're gonna talk later also. They have gait problems, and the gait problem or balance problem can lead to fall and injury. And old age it can be very problematic. All right. So what the primary symptom damage in the brain of Parkinson's disease patient, actually reduction of dopamine in the brain. Again, I think this is actually not the only thing, but this kind of more like the textbook definition of Parkinson's patient. This is like the primary thing we see in Parkinson's disease patient. So dopamine actually is projected from midbrain, from two nuclei, running more like a ventral tegmental area, and substantia nigra, vas compacta, and they project dopamine to different parts of the brain, including the area called the basal ganglia, the prefrontal cortex, also hippocampus and other parts of the cortex, and so forth. But it is believed that dopamine projected to the basal ganglia. So basal ganglia is a big motor control system. So you have reduction of dopamine in this area that leads to the motor problem we see in Parkinson's disease patient. And again, at least this is the standard textbook theory. Probably in real life things are more complex than this. But because of dopamine reduction in the brain, most of the medication given to Parkinson's disease patient are just like the increased dopamine levels in the brain. So we have levodopa is known as a golden medicine for Parkinson's disease patient. It has been around for about 50-60 years, and it managed the symptoms very well. But it comes with some other problems. Every one of this kind of class of medication, they have some other side effects with them. Immuno inhibitors also like an old medication. They have been used also for quite a while, but I think there's some in many Western countries they stopped using them for many kind of also side effects. Dopamine agonists, kind of like the newer kind of medication. Also many different class of different kind of medications. Again, they have a longer half-life, they're more effective, but again, they come up with some problems, which I will talk about some of them later. Some of these problems are hallucinations and impulsivity problems that caused actually by the medication themselves. All right, so as I mentioned, Parkinson, over the last 20-25 years, we know that Parkinson really is not only a motor disorder, it also affects some other processes, including cognitive and psychiatric processes as well. It affects learning, it affects memory, it affects attention, and so forth. And also associated with many different kind of psychiatric issues, including sometimes we see depression in Parkinson's disease patients, and as I mentioned earlier, hallucinations and impulsivity can also happen in Parkinson's disease patients. Again, some of these symptoms actually, what people try to find out whether it's related to the disease itself or actually it's caused by the medication we give to the patients. Okay? Are you guys following? Everything good? All right, so the kind of research I do is the following. We do what we call behavioral neuropsychology research. So we bring patients to university, or we go to test them at the clinic. So basically, so we have like a computerized task. We have like a game which is designed to test certain kind of different kind of behavior or cognitive processes or a motor task. And we try to test the patient on and off medication. And the reason we do this is to try to tease apart the effect of Parkinson itself from the medication we give to Parkinson patients. If the patient are on deep brain stimulation, we test them also on and off deep brain stimulation to see the effect again. Is it caused by the DBS itself, or it's caused by Parkinson itself? Okay. And we build also kind of computational modeling work to understand it's mostly network models or different kind of fatigue models to understand the behavior results we have. To have the results, we need to know what is really causing this in the brain? How can reduction of dopamine is causing certain kind of behaviors? Or we also can, I'm going to talk later about it, some fitting models. We fit the model to data to some behavior, the computational models to find out like which parameter in the model explain the data. And of course, also use a model to have some testable prediction as well. All right. So I'll start talk about like in turn about these projects. We start just explain briefly what reinforcement learning is. So actually, reinforcement learning is actually a very old term in animal theory. The idea is you can actually teach a rat or any other kind of Asian animals, computer or whatever, to do a certain kind of motor response, like based on the kind of the feedback you give them. So you can teach rats to press a lever, say 4 or 5 times, like 5 times. You can only give them like food pellets after you do it a certain number of times, and they learn the motor response very well. That's the main idea. So like the schematic of this would be something like so you have certain kind of stimuli, which lead to certain kind of action. First time, so like you try randomly. So if you have the rat in the cage, you try randomly to get some kind of feedback, and then that kind of the feedback teaches them to do this action again. So that's the main idea about this reinforcement learning model. But the interesting link here is we know that dopamine actually play a role in the teaching signal. So increased dopamine in the brain. There's a lot of recording from monkey brains, rat brains, human neuroimaging studies really show that dopamine plays a key role in learning processes, and in particular for reinforcement learning models. And here's briefly the reinforcement learning models. What's happening is, say, at every time step, you compute what's known as the prediction error. And basically, the prediction error is the value you have of something. You have for everything, we have a certain value, and the kind of rewards you get based on this, and then you subtract them, and it's a prediction error. And then you update, and based on the new reward, you keep updating this. So you update the value based on the feedback you have, based on a certain kind of, so just multiply the prediction error by some certain kind of learning rate, and you update the value of things you're doing. And then you use it's a very standard softmax equation to compute certain kind of responses. You just say if I have to choose between A and B, I basically most likely I choose the one with the highest value. So I can tomorrow I can I don't know, eat X or eat something else, but then usually I like to eat this, so I always so this has a higher value. So this is okay, on a higher value, that's one you usually choose. But you have also the beta parameter, would give it a bit of exploration sometimes. You don't always go for something that the higher value, sometimes you try something different. That's basically the softmax equation for choices. To talk about one project about freezing of gait. I've been working doing this project at my university, IIT Madras, and also Sydney University. Just I'll give you an example of what freezing of gait is. I'll try to, let me see if I can play this video here. Show videos of Parkinson disease patient, and you see what exactly is freeze is. So that's kind of a short freeze here. Sometimes freeze happen when people turn around. I also try to make a turn for it. Sometimes it happens when they start a movement, they start freezing. But after they keep going, it's okay.
So these are patients off medication or on medication?
I think they're on medication, yeah. The issue with actually freezing of gait is, unlike other cluster of motor symptoms in Parkinson disease patient, medication manage it very well. So medication can manage like rigidity and tremor well, but for freezing, for gait problems, medication doesn't do very well. I think they're on medication. For what I've seen so far, yeah. So medication has like almost negligible effect on gait dysfunction in the patient. So here's actually the very interesting. So as you see here, like some of the patient, like the motor function can be okay. Actually, what research has shown actually is it's only when you challenge them that it becomes a problem. So you have a patient, like have a patient walking in an open area, it's completely fine. Once you give them some kind of, okay, like now you make a turn, and then like they start freezing, because they have to switch motor responses. For them, it's kind of like a very difficult thing. I mean, we do it automatically, right? But for them, they have to think, okay, now I was walking, I had to change the motor plan. So it becomes more difficult. Or if they have maneuver some kind of obstacle, they're walking and there's something in front of them, they have to make a different kind of movement. Again, they start freezing. Two different kind of studies showed, like again, it seemed to be not really like a global motor problem, but somehow when you challenge them with doing something else or a cognitive thing or perceptual, you overload them, then it becomes, you can see it more. So freezing gait, it actually we see it more actually in advanced Parkinson disease patient, because Parkinson disease is like a progressive disorder, over these becomes worse and worse, and we see it more in advanced stages of Parkinson. Also, it is different from all kind of motor symptoms. It does not correlate with akinesia or tremor or any other kind of symptoms. And as I mentioned earlier, the effect of medication and deep brain stimulation are not, it's like some reports found it can be effective, some other studies found not really good. So there's like an open work here to really understand what's really happening in freezing of gait and how can you treat it. As I also mentioned earlier, it can lead to falls and injury. Okay. So one study actually we started to focus and to try to study is actually that like doorway width and breathing. One study was done and they found that Parkinson's disease patient will freeze more if they ask them to go through a narrow door than a wide door. Most people, even like unconsciously, you probably will slow down a little bit when you get closer to the door if it's a little bit wide. It's a little bit narrow than wide. But for Parkinson's disease patient, once they get closer, they start freezing. Even though they are told everyone can pass through the door, but they still, for them, they feel they might hit, they don't know what can exactly do, and so forth. So this is actually the study done by a group in the UK. HEC is just for healthy control, and this different color is for the different kind of door size, and this like line in the middle, this line here is the door, and you have the patient walking through this. And as you can see here, for healthy controls, they can slow down a little bit when it comes to a narrow door, but mostly they're going fine. For Parkinson's disease patient, you see a drop, they slow a lot down here when they get closer to the door. And in some cases also that can also trigger freezing in the patients. Okay, so try to use like a reinforcement learning model to understand this. So what we did, we assume with the same model I showed you earlier, we assume that the velocity equate with value. So you have the also value of the entire space, and you have also Parkinson affect dopamine and teaching signal, and you have an agent. Just basically try to navigate through these different doors in the simulation. We have also a narrow door and a wide one. And we assume that basically, if you pass through the door, this is a rewarding thing. You have agent like learning in the beginning, just go randomly in the space. You pass, you get one, and then you learn to go fast. However, in the other case, when you have like a narrow door, you're more likely to hit the walls, and you get some kind of negative signal, which then slow you down. That's basically the idea. And here the model finding, we published this paper, not this year, last year, in frontiers. Here's the experimental data, and here also the computational data, and we see we kind of replicate the finding. Again, here is the computational data. I see also that for head control, again, a little bit of slowing down when we get close to the door. However, when you get in Parkinson patient, you see also a bigger effect when you get closer to the door. But in this also project, we are doing some other things as well. So again, this is experimental and computational project. The experimental part, we see also what other factors can lead to freezing of gait. So one study we are doing now actually is we look at what's called dual tasking, because we have anecdotal evidence that for Parkinson patient, if they are doing more than one thing at the same time, it can actually lead them freezing of gait. So basically, like for you again, if you're walking and at the same time you're eating or you're thinking or something else, it's not gonna affect your movement that much, maybe slightly. But for Parkinson's disease patient, the effect is really big. So we do the experiments, we see when we do dual tasking how that affect their freezing of gait. And also we try to also do the computational modeling as well to understand how it's really working. And as I will mention later, we do also the studies on why DPS is not very effective for gait and what exactly you can do using computational modeling to fix this problem. Any question before I move to the other topic?
I don't know that. I can wait. Is there anybody else?
So as you said that the Parkinson's disease patient are confused, like they have some negative signal while they going while from the small doors.
Yeah.
Is there any effect of height and dip to the patient? Like, do they have some similar kind of thing? Like when they're traveling somewhere on the height.
Okay, I'm not sure if they are like on a high space and they're walking and it might asking if that will trigger freezing phenomena, right? I don't think this was done. However, there's also studies that found that when you induce an anxiety in the patient, they're more likely also to show freezing of gait more. If they didn't make them anxious, it triggered a freezing in the patient. So I would think height might increase an anxiety, and which in turn might probably increase freezing of gait. But I'm not sure if this was done. Probably it wasn't done before. So what is depth? What do you mean by depth?
So for example, if they are standing at the bottom of the mountain and they are looking at the height. So
that I'm not sure. I'm not sure if this was done before or not. But it might be the same thing also. That might increase. I think height might increase an anxiety more than width, I think. But I'm not sure if it was done before or not.
I have one question. You said Parkinson's disease affects the memory.
Correct.
So can you just explain how does it affect? Means in what way it affects the memory, like by learning process going on or?
Okay, so we've published papers before so that actually many studies found actually it affects specifically also short-term memory. Okay. So we see a working memory deficit in Parkinson's disease patients.
Okay, so does it mean that they cannot form memories?
Are you talking about now long-term memory?
No, the short-term memories.
Okay.
The memory formation.
It is actually it's kind of reduced, in the behavioral studies, they still have some kind of elementary, working memory performance. But again, compared to age and education match controls, we see that working memory is reduced in Parkinson's disease patients. So this was done in many studies. So an example, a study called working memory span, how many items you can keep in your working memory. We say a normal healthy subject can maintain up to 7 items or something. In Parkinson's patient, it's a little bit reduced. They still can do working memory, but not as efficiently as an other patient. But also most of the studies found medication actually can help with working memory very well.
Okay. One more question, like you said the main reason for the Parkinson disease is reduction of dopamine production.
Well, I mean, that's like the
But like
spoke different.
but then again you said that effects of dopamine medication is inconclusive. So
No, I mean on gait itself.
on freezing of gait.
An effect on its on gait problems are, we don't know how exactly. I mean, some studies found like it can fix it a little bit, but some studies didn't find any effect. But the effect of dopamine medication on tremor and akinesia, it's mostly like it has been found. It works perfectly well.
Thank you.
We'll ask later.
Okay. This is another project also we did on impulsivity in Parkinson's disease. I'll just tell you briefly what it is. So, again, impulsivity, as I mentioned earlier, is actually caused by the medication you give to Parkinson's disease patient. Sometimes the medication are completely fine. They start a new kind of medication, and then a few days or few weeks after, they start show the symptoms. And usually their partner start complaining, their husband or their wife, they start they start do gambling online, they overuse a credit card to buy things they completely haven't done before, and things like that. So the common symptom within Parkinson, pathological gambling, and also compulsive shopping, hypersexuality. I don't have a picture here for this reason. And also binge eating as well. They sort of overeating, more than usual as well. Again, sometimes we see all of these symptoms or a subset of this in the Parkinson's disease patients. Just briefly, so what's really causing really this kind of symptom? Okay, so we have levodopa for more than 50 years, and actually we know that actually treat Parkinson very well, and we don't have impulsivity symptom. We don't have many reports of impulsivity caused by levodopa. Also in 1970s and later also there are new different kind of dopamine agonists have been around as well, used for Parkinson patients. Again, they didn't actually lead to much, including pregloid and bromocriptine, and have not caused much of impulsivity problem either. It's only 1990s, two drugs. These are the most commonly used drugs now for Parkinson, or at least in the States, in Europe and Australia as well, Pranapixel and Requip. And after these drugs have been used, actually there are dozens and dozens of reports of impulsivity problem. We know that are really linked to this, specifically to this dopamine agonist more than other kind of medication. And also, it's all we don't even know why this is happening. It also happens only in a subset of patients taking this kind of medication. So not anyone on this medication will develop impulsivity problem. So and in this project, we're asking, okay, so what's really causing impulsivity in the patient? And what is the information processing mechanism underlying it? So we did, what is the theory that it could be, so impulsivity can be because of hypersensitivity to what's known as positive outcome, or reward information, or hypersensitivity to negative outcome. So again, the main idea is, say, if someone is alcoholic, so it's one thing, they like alcohol a lot, so alcohol it means a lot for them, so they will always go for it, and even if there's any other options to do. Or it could be that because if you're alcoholic, you're more likely to, you don't have a job, your partner might leave you, and there's certain kind of problems, but all this kind of negative feedback, but that doesn't really change your behavior. You're supposed after you get some kind of negative feedback, you switch your behavior to something different. But if they are hypersensitive to the negative feedback, they keep doing the exact same thing. So we designed a computerized task to look at this. It's known as this. You can either learn from gain or loss. You learn from positive outcome or negative outcome. And in this task, again, the same thing you learn from carrot or stick. You can learn to do something when you get positive feedback. You actually can learn to avoid doing something if you get a negative feedback. To try to tease apart these two kind of learning. And here's a computerized task. Again, if you're a participant, you will be sitting in front of a computer, and this is what you will see on the screen. in the very beginning, you learn just trial and error. I'll ask you for this stimulus, what you want to choose, A or B, and then you say A, then you get some points. If you choose B, you get nothing. For another stimulus, okay, so the one before is actually the positive one. So you learn, like, for this stimulus, I should choose this action to get, to get positive feedback. For the other stimulus here, if you say A, you lose point. If you choose B, you lose nothing. Again, for this stimulus, I should learn to choose B, so I wouldn't lose anything. So basically, by doing this, and we have 4 stimuli of the same thing, and keep subject to do 160 trials, and they keep learning. Of course, first 3 trials, they learn this to do by randomly, they get feedback and kind of change their behavior, and they keep going all the time, and then they see how they learn it. So in this study, so this is like the experimental part. We recruited 4 groups of participants. The first one is, so ICD plus is Parkinson's disease patient who have impulsivity problem. The second group is a Parkinson's disease patient who are also on medication, but they didn't develop impulsivity. The third group, Parkinson's disease patient who are off medication, and the last one is a healthy controls. And this table, like in most experimental study, make sure that people are matched basically on everything. They are matched on education, sex ratio as well, the age. HY is like what's known as Heinon-Yahr measure. It's like about the severity of Parkinson's disease patient, how severe Parkinson is. And here it's about 2.5. It's kind of mild Parkinson's disease patient, early to mild Parkinson's disease patient. UPDRS is like a scale of the motor severity in the patient. Disease duration, they have Parkinson for about 8 to 9 years, BDIs, depression, and so forth. And the most important thing here also, Barrett impulsiveness scale to see how severe their impulsivity is. And as you can see in the first group, Parkinson was patient, they have, as you see from the scale, they have higher number on the scale. It means like they have more impulsivity than the other group. So this is the experimental part of the study. So we have more or less like 20 or 30 plus patient in every group. They all have done the exact this task, and we have the data and reward and punishment. The second part is, so we use reinforcement learning model, but this time is a fitting model. We feed the behavior data to the model, and we extract parameter values to see actually to see a difference among these patient groups. So we feed it to what known as a modified reinforcement learning model known as actor-critic model. Actor-critic model are a class of computational models now used heavily in the basal ganglia literature. And the main idea is the critic, you can think of it as a critic is a teacher, actor is the one that does like the action. So you have them a system, critic that does like the learning, evaluate the signal and so forth, and actor does actions. And you have actually learning happen in both ways. So basically, so you can do an action, you get positive feedback. The critics say, okay, this is very positive. Tell the actor, keep doing this, it's very good. Increase the likelihood of doing the same action. You do a feedback, you do some kind of action, you get negative feedback, the critic tell the actor, this is not good, don't do it again. And so, two interactive system in this way. And actually, a lot of, again, animal work and human neuroimaging studies found there's a different part of the brain are actually represent the critic and actor. Mostly the critic probably is dopamine and the ventral striatum, and actor is like also an area in the basal ganglia and the putamen or dorsal striatum. Again, so apply this computational model based on the data we collected. We get the parameter values and then we correlate with impulsivity measures. So again, the model briefly is the following. So this is the one I showed you earlier, the top figure. But this is model, we also assume that the teaching signal as well can also affect learning about the stimuli. The idea is, so now actually we believe that reinforcement learning, for a long time reinforcement learning has been actually only applied to actions. Like you do a certain kind of a response, it is like you get some kind of positive feedback, you do it again. But actually, it's actually more than this. So also I can now learn also about the stimuli, but also attention learning. If I happen to be able to pay attention to some kind of a stimuli, and this has led to some kind of, eventually to some kind of reward, it enhances attention towards kind of stimuli. Or again, also at the cognitive level, the same thing. If you happen to maintain some kind of information in your memory that happen to be good, then you tend to actually to keep it more and more active. Therefore, reinforcement learning has been shown to apply to different kind of processes, not only motor responses. That makes sense? Again, it's the exact same model with a slight modification. We have the prediction error the same. We have the critic values for the critic. Again, we have two learning parameters. So we have learning positive. If the prediction error is positive, we have a learning rate for it, which is alpha gain C. And if a negative prediction error, we have also another learning parameter. And the exact same also for the actor. Also, you have two different kind of learning rates for positive and negative prediction errors. And choice is the same thing. We have also the same kind of softmax equation for computing the responses. So here in this model, we have 4 free parameters. Again, learning from negative outcome in the critic, learning from positive outcome in the critic, and also the exact same thing also in the actor. So here's the data. So what you see here on the left is actually learning from reward. What you see on the right is learning from loss or punishment in the patient. So this is actually the behavior results. So there's no modeling results here yet. And again, the 4 groups are actually healthy control, Parkinson's disease off medication. The third one, the Parkinson's disease patient without impulsivity, and Parkinson's disease patient with impulsivity. And as actually you can see here is, so this impulsive group, they actually learn even higher than healthy control subject. So you have actually patient. Yeah.
I have a question.
Yeah.
Why is it on meds you have a very high variance? Is that a kind of critical behavior of the model or the data?
Yeah, I don't know. I think just like the data came out like this.
Because
It's
the variance is slightly higher when you see the meds category, right? PD on ICD minus and
You
PD
mean
on
this,
ICD plus.
this group you mean?
Both those groups have slightly higher variance looking.
Yeah, I think it's slight. I don't. Yeah.
Is that the reason saying it's in? If you remove the variances, actually it's statistically insignificant compared to the healthy condition, right?
It is significant. Yeah.
Okay.
So actually, through learning in the Parkinson's disease patient, the variance. I didn't actually think about it. It's a good point. I think the variance could probably be because they are on different kind of medications. Okay. So every medication has different effect. Basically, maybe we are lumping together a whole class of people. When they're off medication, it's become just like a Parkinson's disease patient. But the medication might have, the different kind of medication might have a different effect. Probably that might be the effect.
Curious.
So what actually see here, as you can see here, so learning the impulsive group, we have like the highest reward learning than other groups. And as you can see also on the figure on the right, you can see also that the patient also on impulsive patient, they actually learn way less from punishment than the other group. Okay. So here are the computational modeling results. So first, let's look at the reward learning rate, and we see that again, there's no effect in the reward learning in the critic. In the actor module, again, this actually so what we have done for every we take the subject data from the behavior task, we feed it the model, we get the parameter values. So for every subject, we have 4 values. And we do the same for the entire participant we have, and then we do the averages, and this is what we get at the very end. And for the actor, as you can see here, is we're expecting this to be higher, like the actor would be higher than the non-impulsive patient, but it actually came out non-significant. But as you can see here, that there's actually here more of a medication effect rather than impulsivity effect. So patient on medication in general, there's so higher actor learning rate in general than the other participant. And that's probably in agreement with the issue, like, does medication in general enhance action in general, regarding whether it's you're impulsive or not. We look at the other parameter values. As you can see here, actually, so the one on the right, this is with the one on the right, there's again no effect of learning from punishment in the actor, but actually the interesting thing is learning from negative feedback from punishment in the critic in impulsive patient was much lower than in the other groups.
I haven't read this paper, but I'm curious, comparing this figure to the previous one, can we say, is it, or have you already said that the learning rate in the critic is almost saturated, it's almost similar, whereas the punishment rate in the critic is more significant?
That's exactly the main point, yeah. So this is actually the highlight of the finding. I think this is the main point.
I see.
It's actually seen that patients with impulsivity have actually problem mainly with negative feedback in the critic. So basically, like, if they get negative feedback, it doesn't really change the response. Okay, so that's again the main point I'm trying to say. So this, the problem with impulsivity, it's seen that it's kind of a critic problem. It's not really the action problem. It's seen that at the critic level, the teaching or the evaluative signal in the brain seem to be responsible for the impulsivity problem. And again, there's a lot of shift in psychiatric work and also in psychology and others. The problem, even though it seems to be like, they have they are impulsive, they keep doing this action over and over and over, but it doesn't mean that the problem is at the motor level. It can be actually something preceding this impulsive behavior. Again, the same finding we see here can also apply to other kind of patient population. Many patient have impulsivity problem, OCD, ADHD patient, and of course on drug addiction research as well. The other project we're working on is we look at depression, and also depression happen in roughly 25 to 30% of the patient, maybe higher depending on the report. But actually, we think that we might actually find that the opposite in Parkinson's disease patient. It might be the case that we're thinking about depression and impulsivity are kind of the opposite of each other. We might find the different report, the different finding with depression in comparison to impulsivity. Okay. So the third project, actually, what we have done some years ago, is actually we look at working memory in Parkinson's disease patient. Well, okay. First off, any question before I move to the other? All good now? Okay. So in this task, actually, we used the standard. So working memory is just a term that's kind of a fancy term for short-term memory, like being able to maintain information in your head for a short period of time. And actually, we know now for different kind of literature that short-term memory is actually a key part of many things we do in everyday life. Even though we're not very aware of it. If you think about even language understanding. If I'm talking to you right now, you hear word after a word after a word, right? So you're supposed to keep in your short-term memory the first few words I said, till the last word in the sentence, to be able to make sense of it. So again, short-term memory is key part of many different kind of, even reading a book or some other things also. Short-term memory is a really key part of many different kind of processes. It's also correlated very highly with IQ in many studies have shown that as well. So working memory has been found it's one of the big areas of study right now. And there are also a lot of programs now that have used what's called working memory training, kind of enhance working memory in different kind of population. And it's found that to be actually very good at treating many symptoms in different kind of patients. So we use a task, it's called as AXCPT task, to test working memory in the patient. Again, if there are subject participant in the study, you will see kind of something similar to this. This is an instruction. Really, the main idea is if I see a basically you see a sequence of letters on the screen. In fact, if you see A before X, you press a key which indicate target. But if you see B before X, you press another key which indicate it's non-target. So the key idea is you have to remember what you have seen before X. If you see X, okay, the last time, like a few seconds before, I have seen A. Now I press this or that. This is how we test working memory. Okay. I'll give you a brief an example. Okay, first, like you see A in the screen, and then now you have a delay. You don't see anything, and then you see X, and I ask you. So now, of course, because you remember I've seen A before, then it's target. That's correct. Another try. If you see B, you have a delay, and then you do X. If it is target, this is incorrect. You are supposed to do non-target. And again, so we have different kind of. This is like simple, but you have some other combination of stimuli, and also you keep learning for each one what I have seen before, and then do the right key press just to get reward or not. Make sense? Okay. And in this study, we tested patient again on and off medication. And by that, what we mean by that, so Parkinson's disease patient take medication about either 3 or 4 times a day, morning, afternoon, and evening. So they took their medication in the morning. They came to the lab maybe like an hour or 2 after, and we test them. And the off medication part, we try to withdraw them from medication from about 15 to 18 hours. Basically, we tell the patients not to take the night and the morning medication. So they take that, so the evening, so like, or the afternoon medication, say like, I don't know, 4 p.m. or something, and we wait till they come the next day to the lab to do a test. And actually, you can see a big difference between the patient when they're on off medication. Usually when they're on medication, patient can drive, they can come to the lab, life is okay. When they're off medication, you can see the tremor is like really, really strong. They start shaking more. You can see that this little bit of discomfort, you can see it on their face as well. So we withdraw them from medication for this time, and then we come again to the lab to do testing. Again, we do this just again to tease apart the effect of medication on Parkinson, on behavior and cognition. In this study, we recruited 2 groups of patients, healthy control and Parkinson's disease patient. Again, they match, and the Parkinson's disease patient, we tested them on and off medication. So the one time they come, they're on medication, and a week after they come when they're off medication, or the other way around. We switch the order. Again, it's the main idea. They match on age, education, and so forth. They have been having the disease for about 7 years, and HANR measure said that they have early to mild Parkinson's disease patient. It's not very severe yet. And actually, by the way, when Parkinson becomes very, very severe, I said it's impossible to do any testing with them. When it's severe Parkinson's disease patient, we can see slight dementia problems. They start forgetting things, and it becomes really difficult to understand an instruction and even do this kind of games. So usually we try to recruit patients on an early and mild stage of Parkinson to be able to test them. So here's the result we published some years ago. This starts from the first experiment. And what we found here is, so we have done this task, and we look at the total number of errors, and I think about 72 trials. So as you can see here is Parkinson's disease patient, when they're on medication, actually they even make less errors than healthy control people. The Parkinson's patient off medication, they make a lot of error. So what does it tell you? That when patients are on medication, it can actually help with working memory problem. They sometimes I said can outperform a healthy controlled subject. Just saying, medication actually enhance working memory performance. So one thing I want to say here, that only applies to working memory. We have done some kind of other cognitive measures. With medication doesn't help with everything. In many other cases, medication might have no effect at all on performance, and other cases it can actually worsen performance. Like patients are completely fine on some kind of cognitive measure, but medication makes it worse. So the effect of medication on cognition is mixed. Depends on the cognitive domain you're testing. think, okay, and we used again a modified learning model when we added again user reinforcement learning model, but we have another component and we assume that this is part response. Also, as I mentioned earlier, you can also learn to put information in your working memory based on the dopamine signal, again use a based on reinforcement learning model. I think I'm just gonna skip this because of time. We're just gonna take a long time to explain. But I'll show you the oops. How can I go back here? Okay. So again, here's the experimental results on the left and the model on the right. Also was able to replicate the results with again the reinforcement learning model. Looked at which parameter can explain the data. And actually we found that, again, it's probably what's happening when you give patient medication. When you increase the parameter responsible for prefrontal dopamine, it can actually increase performance in the model. Like in the model can give you an idea which parameter really can lead to the results you have. Can have kind of explanation from the model for the results. So prefrontal dopamine is probably responsible for why we see enhanced working memory performance in Parkinson patients. I think I'll skip this. So all I said briefly. Again, we look at sometimes the tremor, even though tremor is the most common symptom we see in Parkinson's disease patient, it actually not all patients have tremor symptoms, or can be milder in some patients than others. So some patients have actually more severe akinesia symptom, and some other patients have more severe tremor symptoms. So it seems like it's different, even if there are different subgroup of the patient, and they show symptom actually differently. But also we see that working memory deficit are actually stronger in patients with akinesia than patients with tremor. Skip this. I'll talk about, here's the last project. We talk about deep brain stimulation. So I started. Okay, I give briefly what is deep brain stimulation again. Again, you do a surgery, you open the brain, you stick electrodes, and then again, it's a different part of the brain based on the disorder you have. And it has been used for many disorders. For Parkinson's disease patient or other motor disorders, dystonia, Tourette syndrome, you target different part of the basal ganglia. For depression and addiction as well, you target, so for motor disorder, basically you target motor areas. For depression and addiction, you target mode areas in the brain, which is the area called neuroscience. This approach that has been used for many years now. For PTSD, there are now clinical trials. So you think about PTSD as an anxiety problem. They have an exaggerated fear response, and basically you target. There's a clinical trial. They have been targeting amygdala with DBS to treat PTSD symptom. For Alzheimer's disease, you target a memory area in the hippocampus to treat Alzheimer patient. For PTSD, as far as I know, for PTSD and Alzheimer, they are not used yet, but they're still under clinical trials. For the other disorder, they have been already used in many parts of the world.
Is there proof also for addiction?
For addiction, I don't think so. I think it's still under clinical trials, animal work and human trial as well. But again, I'm not on top of the literature for the DBS for other disorders. I'm probably 100% sure. But for OCD, for depression, yes. For Parkinson and other disorders, that's been used. I can't recall the place in Kerala that does the DBS.
There is Sri Chitra.
What's that?
There is Sri Chitra, which is close to us.
Okay.
Trivandrum. They do a little bit of DBS.
That's it. Yeah,
for Parkinson's.
So this study we did many years ago. We looked at it's a computational modeling study, and also experimental study. We tested Parkinson patient on and off deep brain stimulation. And this was in 2006, 2007. This was my first interaction with patient on deep brain stimulation. Again, we do DBS again with medication. We give to Parkinson. Over the many years, they cause many problems. Because sometimes called like dystonia, and they can become toxic and things like that. So you can't really keep living on medication forever. You have to either stop it or minimize the medication. So patient will then switch to deep brain stimulation, which basically target an area in the brain in the basal ganglia known as the subthalamic nucleus, which is like a very, very small, it's a peanut-sized nucleus in the brain, and you target this with deep brain stimulation. Again, this was my first interaction working with patient. It's actually amazing, actually. And then, so what you see here is actually the battery, and it has like a different kind of parameters where you can program the kind of the pulse width, the frequency, and other parameters of deep brain stimulation. And that can also change it based on the patient and also over time. So every patient, you kind of try a different kind of frequency for them to see which one will work best to manage your tremor and so forth. And the same, and also you have a control, remote control, you can also try to increase and decrease and so forth. And something you give to the patient, you give it limited, control over these parameters to change it, just to make sure you can manage your symptoms well. For this study, actually, we turn it off just to see what exactly happened to the patient after they're off DBS for a while. It's actually really magical because the patient, once you turn it off, a few seconds after, patient their tremor was going just crazy. Like they were tremoring, they're shaking all the time. Their legs and their hands was and the frequency actually was just really strong. But in this certain study, what we have done is we looked at we know that again DBS actually works very well, but it comes at a price. We know that it causes also impulsivity problem in the patients. So the task developed then by my supervisor then, Michael Frank, we look at exactly how we look at impulsivity in this patient. So we have this task, which basically is the following. The task has two different conditions. One is high conflict versus low conflict condition. The high conflict one say, if I give you the choice between two different things, maybe to eat chapati or paratha. That's my problem every day. Which one to choose? So that's high conflict one. Both are really, really good, and it's hard to make a decision. Or he's just go to Paris or go to Rome. That's high conflict. You don't know how to decide this. I think for Sham, he'll probably go to Rome. And low conflict condition, just to go to Paris or on vacation, or I don't know, to write a grant. This is the easy one, This is for this kind of condition. Usually, in the high conflict, people usually take longer time to decide, so you don't know which one to choose. But for the low conflict one, it's always an easy decision to make. And the task by Michael Frank, again my supervisor, then we train people. So how to do this in the lab? So we train people with different stimuli, and everyone has a different reward value. So with the stimulus in the left side has 80% reward value. If you keep choosing that, you get usually get reward. The second one, 70%, 30 and 20. And basically we train on all of this stimuli to learn their value. And then the main idea is that we test you the one was have high value, with 80 versus 70, and to see how we choose them. Actually, the very interesting thing here, all the subjects are not aware of the training before. They just keep learning and learning, but if you ask them at the very end, you don't have awareness of why they take longer time to decide. It becomes just kind of a very habitual kind of response, and they don't know the answer. And the conflict one, again, the 80 versus 20% reward is easier. So that's very easy. You take the one with the high reward value.
These are visual cues? Are they visual cues?
Yeah, just using the stimuli in the screen, and they learn. And here are the results. Here we have 5 patient groups. The first one, seniors, this is healthy controls. And then the other 4 are different kind of Parkinson patient. So Parkinson patient off medication, and then Parkinson patient on medication. The last 2 Parkinson patient off DBS, and then Parkinson patient on DBS. So as you can see here in the head, for most people, you can see that for the high conflict, you take more reaction time to decide. So reaction time is higher, you take longer time to make a decision which one to choose. for in the healthy control, you take more time in high conflict than low conflict. The same for off medication, for on medication patients, for off DBS, but not on DBS. When patients are on deep brain stimulation, they decide very quickly. Again, that's kind of like like a task that show proof that of the anecdotal evidence that really patient have impulsivity problem. This can happen. This is kind of like it's good to have a task. Sometimes like when you use questionnaires, you can get different results. You don't know what's really happening. This would have been used before then. So the next thing we have been working on, actually just start to build a basic ganglia model to look exactly why exactly. So this is a schematic of the basic ganglia, the cortex interaction with the basal ganglia, and we'll try to see why actually applying deep brain stimulation can actually cause impulsivity. But not only that, what can you do? So a good thing about computational modeling is not only replicate existing results, but also I can play with the model and see what parameter I can do that I'm able to actually minimize the problem. So when I maximize efficacy, I can treat tremor and all other problems using modeling, but at the same time also like to minimize side effects. What's actually happening now in this case is just this patient that the neurologist, like you again, trial and error. There's no methodology behind what exactly to do. Okay, let's like minimize the frequency, let's increase this, let's decrease that, until you get rid of the problem. But there's no algorithm or method that tell you how exactly I can deal with the problem. But again, computational neuroscience work here has the potential to be able to deal with this kind of problem. You can do the simulation, you can simulate impulsivity in the model, and then you can find treatment or potential treatment for this kind of symptom using the computational modeling. Okay, the summary of, or maybe if you have any question about deep brain stimulation, before. Okay. Anyway, so again, the summary of my lecture today is actually we know that actually Parkinson's disease patient, so is even though it is usually defined as a motor disorder, but again we see cognitive problems, working memory, learning, attention problem. It also, as I mentioned, affect also psychiatric processes. We see depression in the patient, we see hallucinations, we see impulsivity, and so forth. And we can use computational modeling to actually to understand how medication works, but also to come up with testable prediction as well. Thank you. So I think we have time for questions and comments. I never got any questions from that part.
Because some of them will interact with you anyway.
Okay.
Yeah. I have a question probably of common use. I'm going to ask
Sure.
it. When we talk about substantia nigra,
Yeah.
PD, and we're looking at levodopa, there have been problems with levodopa after 3 months of use. How do the patients actually manage it? For 3 months it works, then there is a year and a half of non-functioning levodopa kind of drugs. Levodopa, for example, has been known to non-function after a particular period of testing. So how do you compensate for that? For example, I see that some of the patients are like 7 years plus on medication.
Yeah. So actually, there are at least two different kind of problems with levodopa. First, levodopa have a shorter half-life than dopamine agonist. So like I'm blanking the term. Yeah. So they wear off quickly. So you take medication, after 3-4 hours, it's wearing off. You have to take another. You have to take something else to keep you to manage the motor symptoms. Dopamine agonists have a longer half-life. They can stay in the body longer. You can manage it for a longer period of time before even you take the second dose. Levodopa have been known for this kind of problem. You don't stay longer in the body to manage the symptoms. Other problem as well. One of the most famous problems with levodopa is what's known as levodopa-induced dyskinesia.
Mm-hmm.
But it's happened over many years. You take levodopa for 5 to 8 to 10 years or so, then you develop dyskinesia problem. Dyskinesia basically is like you start like moving like your hand and legs uncontrollably. So just like you keep moving like this and your hand keep moving all the time. Kind of actually the opposite of you move from a hypokinetic state to a hyperkinetic state. And the way you deal with this, so again there are the thing also about this practice, different people do different things. It's almost like a philosophy. Everyone has like a different view, and there's no one way of doing things. So what people do usually, either you take the patient off levodopa, put them in dopamine agonist, maybe minimize or most like reduce the dose of levodopa, some of the patient, or add a different kind of medication to it. So if you're taking levodopa, you reduce the dose and add MO inhibitor and add dopamine agonist. Pretty much most of the patients in the Western world are on multiple kind of medications. It's just rare to find someone on one kind of medication now. It's like, you reduce one, you put the other one, it causes problem, you reduce and maybe add another thing. And so, and it seems that our dopamine agonists have their own kind of problems, but they manage with this levodopa-induced dyskinesia and wearing off problems.
And what kind of knockout models would be appropriate for these kind of studies? I mean, I'm talking about rats and mice.
The most common rat studies, so there's one called 6-hydroxydopamine.
Okay.
It's a neurotoxin. I've never done animal work. Even I've interacted with animal people before. And it sounds funny too. So for rats study, they usually use like 6-hydroxydopamine. It was a neurotoxin. It's known actually to kill dopamine cells. Mm-hmm. That's a common rat model of Parkinson. For monkey work, the people do MPTP. Okay. MPTP is also like a neurotoxin. And I don't know why MPTP is used for monkeys and 6-hydroxydopamine used for rats, but this has been what is commonly used in the literature. There are many different kind of genetic models and other things, but these are the most commonly used like neurotoxin for Parkinson model in the animal literature. And actually I've seen visited labs and they do MPTP research on monkeys, and they look at the monkey, it's like they really Parkinsonian. They cannot really move their hands. And so whereas one thing, you don't usually see tremor in the monkey. It's just hard to develop. I think it's based on the monkey species, I think. But the akinesia and the stiffness and all that, you really see it after on MPTP. I think just only for like 3 or 4 shots, like over a week, you can develop a Parkinsonian model of monkey model of Parkinson.
MPTP is Langurian or rhesus?
What, sorry?
Which kind of monkey are we talking about? MPTP
I
is
think macaque, yeah.
Macaque.
Yeah. But again, I think that's the most commonly used. I think it's also used with green monkey and other monkey species as well.
And none of them show tremors, usually?
I think in green monkeys, I think you can see a bit of tremor. And I think other monkey species, I don't think so. But I mean, no one really knows why this is happening there.
Any other questions? Anybody else?
So in the case of Parkinson's disease, like when it starts, when we got to know that the patient has Parkinson's disease, are there any levels like the effect of disease increases as the age increase?
As what, sorry?
For example, the freezing, is it like increase as the age increase, something like this? The effect.
Decrease. Freezing decrease?
No, increase, I'm to say. As the age increase, the effect of the particular disease increases. Is it something like this?
Well, honestly, I'm not sure if it's an age effect, but I think I'm not 100% sure if it's age related, or it's like the severity of Parkinson itself is related. But also these are like a kind of two combined things. Like, usually, Parkinson again is progressive disorder, becomes worse over time. Parkinson is more common in older age, so I'm not sure if anyone has really teased apart this effect of its really age, of the severity of Parkinson itself. That's the main factor underlying freezing.
So on the basis of what we give them medication?
What's that?
On the basis of what we give the amount of medication, like how much the dose of medication is given in the basic.
This is not very scientific. I think it's kind of art. I don't think patient usually, again, it's trial and error. And medication doesn't really manage very well for advanced stage Parkinson patient. You start with a normal dose, and you see the patient how they feel, and you can do some kind of simple motor test. You can look at motor coordination, motor timing, their walking. If it works fine, if not, you come back after 2 weeks to the clinic, and you just increase the medication, add something else. It's just like, it's all try and error. By increasing the dose, adding medication, changing the medication, there's many things you can do until you're able to manage it. This was happening in the neurological clinics and real practice.
Is there anybody else with questions? Any kind of...
You seem to want to ask a question. You seem like... Go ahead.
So you were saying that there are long-term side effects for the drugs. So what are they? For each of these drugs, does this vary? Side
each
effects.
of these drugs? What? Say it again?
Side effects. You were saying that there are side effects. So does this vary with each drug, or
yes.
what are they?
So it's known that levodopa have long-term effect. They cause dyskinesia. So we see it actually more than in dopamine agonists and even MO inhibitors medication. And honestly, no one knows really why. There are different theories and different findings, but we know that levodopa, you take levodopa for a long time, you develop some kind of problems. We don't see that again with dopamine. So the problem with dopamine agonists, as I mentioned, the impulsivity part. But this can happen actually right away. You take the medication, a month or two after you start seeing the symptoms. So it does, it varies by medication. Yeah.
Anybody else? So if that is the case,
Sure.
thank you.
Thank you very much.
Talk, and we'll catch up in the afternoon, probably in the lab with the follow-up questions.
Thank you very much. All right.